spent 25 years on this topic, and here's an hour, right? So what am I going to say in an hour? There's lots of things that are in this field. Most famously, the evolution of virulence. Why are some pathogens more nasty than others, and how will they change when we change their world? There are very interesting issues to do with the evolution of emergent diseases. What happens when they first come into human populations or animal populations? There are very interesting questions about the evolution of the life histories, and from what Steve was saying yesterday, there are analogous issues with infectious diseases. Why are some diseases very acute and some chronic? Why are some highly infectious and others less infectious but for longer periods of time? Why do some make you sick before they become infectious and some make you sick after they become infectious? What determines the difference in times between those? That whole area is, I think, going to be one of the most exciting areas in the future. Very difficult, but people are making progress on that. And much as I'd like to talk about all that side of things, in a given an hour, I thought what I'd do is actually talk about things that are in my head, because that way I'll have more fun for the rest of the week. But my bottom lines are these. First off, I think the golden era of magic bullets, and particularly magic drugs and magic vaccines, that's over. And we're now on the hard stuff. The real science, the real evolution sets in this century. We were lucky last century. The hard work is going to happen this century. I want to make in this talk these sort of summary bullet points that a whole lot of evolution can take place, and it can take off in different directions. And it's actually very hard at the moment to know when something's going to go in one direction or another. That vaccine-driven evolution can happen, and it's about more than antigens. It comes to the evolution of drug resistance, that it's a field dominated, I think, by a lot of folklore and wisdom handed down. It's eminence-based research handed down from wise people to wise people for 60, 80 years. And when you actually dig into the literature, the evidence base for what's going on is often very poor, and the conceptual basis of what's being done is even poorer. So for me, the big challenge in this century is going to be to advance the state of the science behind drug resistance management to the level that it is in other areas of evolutionary biology. And if I can get one message across, is there's going to be no simple rules here. There's a tendency for people to want to say, you know, this always works, or this is always good, or here's a rule we can apply in a whole lot of different situations. And that's just madness. It's like saying, is there a cure for cancer? You know, there isn't a cure for cancer. There's a whole lot of different cancers. There's going to be different ways of doing it. There's no simple cure for drug-resistant pathogens, no simple way to make vaccines. Complexity is everywhere, and simple rules should be out. And I know that's unpopular, but that's the reality in my view. Okay, so you all know the drug resistance evolution is a significant problem. This is the story in malaria. Chloroquine was introduced at the end of the Second World War. Chloroquine resistance seen 10 years later. Progranol resistance, pyrimethamine resistance, and so forth. And this is the way it's run for the last century and is now running in this century. So artemisinin resistance looks like it's starting to appear in Southeast Asia. Artemisinin is currently the last frontline drug available for malaria. So at the moment, unless something pops out of the pipeline, there is no plan B. The WHO, the World Health Organization, now has a policy that it expects drugs to fail. Okay, so the WHO has as policy that drugs will fail. And so the expectation is we need to keep a pipeline like this going of drug discovery for as long as there are parasites around. And you all know that this sort of evolution plays out everywhere. The story is even worse in TB at the moment. There's now strains of extensively drug-resistant TB that aren't treatable by known drugs at the moment. And I'm guessing that Bob's going to say something about the situation in American hospitals. This evolution is everywhere, and it's pretty gruesome. And I think the most worrying part about it is that this plan we have of endless discovery of new drugs, that there'll be something around the corner, it's not clear that is actually going to work in the long run. The main reason for concern, I think, is that the economics of this are very poor. Market forces are not in favor of developing new drugs which you will protect, use only when necessary, restrict to professionals in the hospitals. The market forces don't work to solve this problem. In fact, it might be a bit better in malaria, where the WHO can get together with Bill Gates and the G8 and start defining a market, describing a market, setting targets, and offering to pay for millions of doses of drugs. And under those situations, that sort of artificially constructed scenario, market forces might help keep this going, although it's extremely politically unstable, requires that the world cooperate. In the case of the rich countries, where it's entirely down to market forces, it isn't clear to me it can work. And worse than the actual drivers is that every next generation drug is more expensive than the last. There's never been a case where it gets cheaper. Oops, we didn't discover the cheap one somewhere down here, which we magically just turned up. And then the other question is, how many drug targets are there that can be worked up? Is there an indefinite supply? Yeah, to me, the reliance on the pipeline is kind of nerve-wracking. So the aim of the game for the evolutionary biologist and the evolutionary medicine specialist is try and slow down the rate at which these things fail. And you notice there are interesting differences in the time from release of the drug to resistance first being seen in the field. And these big differences, chloroquine was 10 years, some of the others more or less instantly, those are the sort of things we would like to know about. And just because resistance has appeared in the field doesn't mean that that drug is useless internationally. It takes time for that resistance to spread around the world. That's the sort of thing we want to slow down. And in the second half of what I'm going to say, I'm going to talk about some of the thinking that's going on about how we might do that. What I want to do to begin with, though, is talk about the sort of opposite of that evolutionary story, which is the equivalent picture for vaccines. And these are the childhood diseases for which we've had very good vaccines, often for many, many decades or even over a century. And we didn't see evolution. In fact, we managed to eradicate smallpox, and we managed to eradicate polio from very large chunks of the world, and maybe we're going to squeeze it out from the whole world. And so we had millions and millions of people around now. Many of you are here because of this triumph. This has been great, and there hasn't been any evolution. And this is a very, very different picture from the drug case. Anybody want to explain that? Why is this picture so different from the drugs? So the suggestion was the difference between virus and bacteria. Okay, that's one interesting possibility. Population size of the organisms, okay. Yeah. So how would that, just explain that a little bit more? So that was the immune system evolves. Oh, okay, so there are antibodies around anyway, yeah. This is prevention as opposed to treatment. We eliminated breeding opportunities.
Yeah, it's about transmission. So with the vaccines, you're curbing transmission, whereas a lot of the drugs that we give malaria don't actually stop transmission, so they can continue to transmit.
Anybody else want to?
Can't go fast enough.
Yeah, I'm really glad I asked this question. So that is the first time somebody said something I've heard before. So all the other suggestions I've never had before. So let's keep this up a little bit longer in case there's some more I haven't heard.
Maybe coevolution with viruses is different than with bacteria?
Uh-huh.
Less strength of selection in this case. The selection force is a lot less strong.
Well, except that some of these vaccines generate lifelong immunity, so they completely clear any pathogen that gets into those hosts.
I guess, I mean...
But you're right, in principle, there could be different strengths of selection, no question.
Right.
To clarify the point, am I confused? Or is pertussis a bacteria?
Right, I actually do think pertussis is very instructive because pertussis is one for which there's evolution happening. The vaccine is in some trouble at the moment, and it's extremely controversial about why it's in trouble. So we could come back to bacteria, to the pertussis case. But it's true the others are all viruses, for which drugs, broadly speaking, don't work. Obviously, there are some exceptions, but HIV, obviously. But yeah, I think this difference is actually very, very interesting. And it's barely commented on, and we don't actually understand this. I've got ideas, but we don't have a very good understanding of why this evolution, leading to the eradication of a disease. So this disease, this virus, failed completely to evolve to the point that it went extinct. Colour? This will be my last one.
I see you don't have flu on this.
So flu vaccine actually, the flu vaccine fails, but not because of anything that the flu vaccine is doing. The coverage of flu vaccine is very low and almost negligible selection pressure. So the immunity is constantly being undermined in the flu case by the evolution of the virus, but it's natural immunity. The vaccine itself is imposing almost nothing in that case. I don't know about you guys, but at Penn State, where the university gives out the vaccine for free, it makes it very, very difficult not to get the vaccine. All sorts of clinics around the corner, they get coverage about 40% of the highly educated people in town who are right next to Penn State, got Penn State insurance, etc. So 40% is almost no selection pressure. So I don't think of the flu vaccine as actually being a terribly interesting vaccine. Ben? Yeah, so that most of these are unique to humans, not zoonotic. That is also true, though, of a fair few of the, like the malaria case, for example. They are human-specific as well. So this remains a unexplained phenomenon. One of the consequences of this phenomenon, though, is that by and large, medicine has not been terribly concerned about the prospects of vaccine-driven evolution. And that's because the history suggests it's not a problem. I want to show you some cases where, in fact, vaccine-driven evolution does happen. My theory on why it didn't happen in these particular cases is these are all acute childhood infections. They specialize on fully naive children, usually ones when they first pitch up at school. So for some reason that we don't fully understand, these things naturally were surviving and existing in the absence of immunity. So as soon as a person gets measles naturally, they can't get measles again, and they have lifelong immunity to measles. So for some reason, the disease has boxed itself in to operating only in fully susceptible individuals. And it can't operate in a vaccinated, an immunized population. If you vaccinate, all you do is shift the cause of that immunity from natural infection through to vaccination, and it's just the same world from the point of view of the pathogen. It still can't get into that immunity. Think about those big measles epidemics that you see, the sort of classic ones. Every time there's a measles outbreak, spreads through a big population, it dies out. It dies out because there's huge numbers of immune individuals and they're not enough to sustain the disease.
Andrew, is it possible that something is going on with the population, the effective population size being smaller because you're not getting selection within individuals for resistance? It's really just sort of between individuals?
That's an interesting thought, which I would have to give some more thought to. Possibly. Yeah, so the childhood diseases are specializing on these young, naive individuals, and so natural immunity was somehow evolution-proof for these diseases. This intense selection at the end of every measles epidemic for any measles viral strain that could get into an immune person. And that intense selection happened every epidemic. So every 3-4 years, there would be intense selection for immune evasion, and they couldn't do it. So there's something about these diseases which makes them specialize on young kids and exist only in those fully susceptible ones. And natural immunity is, for some very interesting reason there, evolution-proof. And that's why we call these things childhood diseases. That's why they are circulating amongst the kids, because of that property. And it's a very interesting question why they have that property, for which nobody understands. And I would say that is one of the squillion-dollar questions in evolution of infectious diseases. Why are these things so incapable of getting through immunity when clearly all the diseases we care about these days, HIV, malaria, hepatitis B, C, all of those sort of things, are very good at evading some types of immunity getting around and going on? Yeah, so pertussis is a very, maybe we'll come back to the pertussis case in discussion. Don't let me forget that. I'm just a bit conscious of the time. You're looking worried there.
So kids can get measles too. Sorry, malaria too, I assume.
Yeah. Yeah, that's right. So the question was, children get malaria too, so what's the difference here? Yeah, in the case of malaria, sure, kids get them, and that's where most of the death is, but people could repeatedly get malaria parasites if they're exposed for the rest of their life. So the immunity that develops for malaria is weak, partial, mostly anti-disease, not anti-pathogen, and it's not this one-shot lifelong immunity like measles. Get it once. If you get a good infection and generates good response, that's it. You'll never get measles again. Yeah, yeah. So probably people in the room understand that situation better than me, but that's a situation where the virus goes latent in the body and comes out later in life. So that is not a reinfection scenario. Yeah, and I think it's very early, the chickenpox scenario, actually. I think, you know, the widespread coverage of chickenpox vaccine, that's a reasonably recent thing. Okay, so what I want to leave you with at this point is that is a very interesting issue in contrast to the drug resistance scenario where evolution dominates the situation. But that is a rather special history. That is of the diseases we didn't need immunology to develop vaccines for. An idiot could make a vaccine against these things by grinding up bits of this stuff and sticking it in people. Okay, it doesn't need anything fancy because, for reasons we don't understand, those diseases are very easy to generate lifelong immunity against, with the exception of the pertussis, which I do want to come back to. So I don't want you to think that the past is any predictor of the future on the vaccine situation. Okay, we've been lucky so far in humanity with what we got on the easy stuff we've done, and now we're working on the hard stuff. There are people that think about the evolution of pathogens in response to vaccines, prompted, I think, largely by the flu scenario. People have in their head that if there is a person has seen a particular variant of a disease, they become immune to that, but there's another variant of the disease which can break through that immunity. So if you're going to vaccinate with strain A, then strain B will have some success in those people, allowing strain A to flourish. And that's this idea of escape variants, or various different names associated with that, but strain replacement or what have you. And this is a particular molecule that's involved in quite a few of the candidate malaria antigens, vaccines, and the claim is this thing sort of hangs out on the end of the red cell, and some mutation somewhere in this will change the structure so that the antibodies that bind to that thing no longer bind as well, and therefore that parasite won't be as inhibited, and it will have an evolutionary advantage and spread in the population. So that mutant structure will have an advantage that spreads, and that is this will cause the erosion of the vaccine efficacy. So what is a fantastic vaccine to start with will become less fantastic in due course. And if we can't do something about that, that could in the limit lead to the vaccine becoming completely useless. That's like drug resistance. So it erodes what was a fantastic tool away to nothing. So to the extent that people think about vaccine-driven evolution, this is the way to think about it. And for example, in pneumococcal vaccines, this is a concern that you'll get strain replacement. In HPV, cervical cancer vaccines, the concern there is there'll be strain replacement and so on. And the flu situation is of this sort. Were we to get very high flu vaccine coverage, it would likely generate selection for variants that aren't in part of the vaccine, and those would spread. And that's very important evolution, but it's not what I've been thinking a lot about, because to me it's kind of obvious. The one I've been thinking about more is whether or not one could also, or as well, or instead, get the evolution of virulence, more virulent strains. I mean virulence here. So virulence is one of these words in this business which means a lot of things to different people. The plant people, the vets, the medics, the evolutionary biologists always have different views on what is meant here. I mean it in the sense of hot and cold strains. Strains that cause more or less severe disease. So all else being equal, strains that on average cause people to be more sick or die faster than other strains. And so for a number of years now, we've been interested in the question of whether or not some types of vaccines might provide an environment within which hot strains can spread, leading to replacement of less virulent strains with more virulent strains. And in retrospect, this was kind of obvious from some of our earlier work on rodent malarias, where we've been working on the evolution of virulence and looking for fitness advantages to virulence, and why is malaria so nasty was a question I started on at the beginning of my career. And one of the things we noticed early on with our work with rodent malarias was that the more virulent strains were harder for the mouse to clear. So they persisted longer in the mouse. The immunity struggled more with the virulent strains than with the less virulent strains. And this is after all the action has happened. So the mouse has lived or died and it's got things under control and it's clearing them out. It struggled to clear them out if they were virulent parasites. So the ones that had made them most sick early on caused them the most difficulty clearing out. I didn't think all that much of it at the time, which seems kind of interesting in retrospect now. But it does make, in retrospect you think, well, maybe that means that immunity is going to bear down disproportionately on the nicer strains, and the nastier strains are going to have a relative fitness advantage and be able to spread in the population. And so that's what we've been working on for quite some time now. I'll show you just a couple of our experiments which I think point in that sort of a direction. We've been working with this thing called AMA1, which is, you don't really need to know the details, this is the parasite when it's in the bloodstream looking for a new red cell to invade, and on the surface it has the binding part to the red cell. It has this protein, this thing called AMA1, and it was discovered in the 1970s and 80s that if you could make antibodies to AMA1, it prevented the red cell being invaded. And the nice thing about that is that there's, because of all the vaccine experimentation, a large amount of this AMA1 is available, or at least many different types are available, including for the species of rodent malarias that we work on. And these antigens are recombinant antigens, so they are stuck into bacteria or yeast, the genes, and then the product of those is taken as the vaccine. So that means that you can get extraordinarily highly purified versions of this molecule and just inject that in and get an immune response, an antibody response, which protects against invasion. And from my point of view as an evolutionary biologist, it's really lovely because we know precisely what selection pressure we're imposing on the parasites because we just stuck that antigen in. So we know exactly what's going on. And this is one of the experiments that we've been doing. This is one that Katrina and Vicky were involved in. What we do is we passage our parasites and we make them more virulent by serial passage. That's pretty standard practice. It's true in almost everything, that if you passage a parasite through something, it will become more virulent to the thing you passaged it through. And so we can make hot or cold strains just by that serial passage approach. And this is an example of the parasite density through time in naive mice, so not vaccinated mice that have been infected with the ancestor, that's this one here, or the derived virulent one, that's this one here. And you can see it grows a little bit better, gets a little bit higher, but there's that clearance issue. It takes a little bit longer for the mouse to get the thing under control and get rid of it. So that's what happens in unvaccinated mice. And then you vaccinate with this AMA1, and you get very good control of the avirulent strains, but you can't control the virulent strains in the same sort of way. So these virulence, this is actually peaking a little bit higher than it did in unvaccinated hosts. But the difference here is the key thing. There's a very big advantage, at least in terms of within-host growth, and I could argue the things that are associated with transmission, for the virulent strains in these hosts, these immunized hosts. So by vaccinating the mice, we can change the relative fitness of the virulent and avirulent strains, and the virulent strains have a disproportionate advantage. Okay, now we don't actually know the mechanism for that. My assumption is that these things are more virulent, they're growing more rapidly, they're more aggressive parasites, and that the particular antibody titers struggle to control them, and so it takes a while for the body to ramp up more antibody production and get things under control. But in that time, the virulent parasites have this fitness advantage and could spread on through the population. Now we just finished, this paper came out last week, and this is a heroic experiment that Vicky did, where we actually took a single parasite and then we grew it up and evolved it through lines of mice that were just vaccinated with the adjuvant and lines of mice that are vaccinated with this AMA1 antigen. And this, I have to say, I look at this picture, to me it's one of the iconic pictures to emerge from my lab. This is what happens when you passage these parasites through naive mice. It's very easy. Every line more or less continues. We get the odd accidental loss. But each of these dots is a mouse, and we go mouse to mouse to mouse to mouse to mouse, and all is pretty easy. In the case of the vaccinated mice, we've often failed to get enough parasites to passage them on, because the vaccine works really well. So the vaccine suppresses the parasite densities, we struggle to get them out and pass them on, and so we have this endless loss of lines. And you can see we struggled to keep the damn things going. We really struggled. And then around here, there's a blossoming, and we started to be able to keep these lines going quite easily. So this is the sort of selection one imagines in a parasite population out there, vaccinated hosts. Most of the time the vaccine works pretty well, and then through time it starts to get less and less effective until it's pretty easy to keep these things going. And that's the vaccine efficacy eroding away. The vaccine gets useless in 20 generations.
So is the infection on the bottom more virulent than the infection on the top?
Yeah, I'm just coming to that. Yeah.
How long is the time interval?
Oh, so we did this every week. We did this weekly. Yeah, and that's a good question because it's an arbitrary choice of what time we do the transmissions. You have to decide something, and so we did once a week because that meant Wednesday was transmission day.
With the serial transmission of malaria, do you think there's going to be any difference? Because usually they go through a sexual
Yeah.
stage.
So that's a very expensive question to answer. We could pick that up again. I mean, I really don't have any answer to that. We have transmitted the lines through mosquitoes, and the virulence differences are stable through transmission, is all I can say. We have never done experimental evolution through mouse, mosquito, mouse, mosquito, mouse, mosquito. And I really, you know, if I had the right case, a nutcase postdoc came along, actually it might be 2 or 3 nutcase postdocs. Okay, the key thing is that in all of this evolution, the target sequence in the parasites did not evolve. We know precisely the antigen we put in, and the antigen did not evolve. No evolution at the target site. None of that fancy mutations in these structures, nothing happened to the target site. So that was not antigen evolution. So what we did was to take parasites at the generation 10 and parasites at the end of the experiment and assay the virulence, and this is what that looks like. So this is anemia in the parasites that had gone through the naive mice or through the vaccinated mice, and in this case, after 21 passages, we also compared it to the ancestor. And so the lower down here, the more anemic the mice are, and you can see that the longer it went through vaccinated mice, then the more anemic it was, less so than the evolving through the sham vaccinated ones. So when you take these parasites and stick them into naive mice, they cause more severe disease. So we created parasites by passaging through vaccinated mice that cause more severe disease in non-vaccinated individuals. So, summarizing that, we can easily find and create these strains that cause more virulence. And those are the ones that get through vaccine-induced immunity. There is another reason why vaccines could create immunity, create greater virulence, and I want to do this pretty quickly. If you ask anybody, why is it that pathogens aren't super hot? What is it that stops really, really hot pathogens like, you know, Ebola type things, instant kill, very, very fast kill, high case fatality rates? What stops those spreading in the human population? And the argument usually is, well, they kill their host and therefore themselves. They don't have any evolutionary future. Okay? If that's true, what happens when you keep people alive with a vaccine? And this is an argument that we made some years ago now. If you had a malaria parasite in a kid that killed the kid, that parasite, if it was so nasty that it killed the kid, it would be removed from the population. If you vaccinate the population, keep it alive, those parasites can persist in the population and spread on. So if the vaccine is leaky, if the parasites come out of that vaccine, they can spread on. So that means that by keeping the people alive, which is the aim of the game, of course, we also keep the more virulent strains around, and those can circulate in the population in a way that they couldn't have done before. And we made that argument mathematically some years ago, and here's some of the math, and other people have done this, and, you know, some people find it reassuring that there are equations there. To me, the issue is more a question of experimental scenario. Maybe I'll skip this. Experimental scenario, and that's very hard to do in people. And so for some years now, I've been working on Marek's disease in chickens. I have to say, I think the farm animal situation has immense capacity to advance the science in these sorts of areas, because you can do the experiments that you can't do on people, because a lot of this requires that we deliberately infect individuals. And that is possible in the farm animal context. The other thing is